In this section, we are going to discuss the superantigen stimulation. How there is difference of stimulation of antigen and superantigen. Do you know what is superantigen? Well, superantigens are mainly bacterial products, for example Staphylococcus enterotoxins or viral proteins, that induce non-selective T cell activation. The response of T cell to superantigen is not clonal. Clonal means a large number of T cell are activated, while a highly specific interaction of processed peptide embedded in MHC complex with alpha chain and beta chain of the T cell receptor. This interaction is mandatory during antigen-specific T cell activation. But remember that superantigens don't require intracellular processing. Superantigen bind as whole protein to the outer side of variable region of beta chain of T cell receptor. Around 100 times more T cell react with superantigens than with normal antigens. Some superantigen induce activation of up to 10% of peripheral blood T cell. On superantigen activation, CD4 plus T cell release a high amount of cytokines, which can mediate toxic effects in the host. The pyogenic toxin superantigen family can cause food poisoning, fever, vomiting, and diarrhea, and include Staphylococcus aureus enterotoxins, staphylococcal toxic shock syndrome toxin 1, streptococcal superantigen, and several streptococcal pyogenic exotoxins. Although these molecules all have a similar structure, they stimulate T cells bearing different variable region of beta chain of T cell receptor. They are strongly mitogenic, which means they stimulate mitosis for these T cells in the presence of MHC class II expressing cells. Staphylococcus aureus enterotoxins is one of the most potent T cell mitogens known, causing dramatic proliferation in the concentration range of 10 raised to the power -13 to 10 raised to the power -10 million. Like the other superantigens, it can cause a cytokine storm, involving the release of excessive amounts of interleukin-2, interferon gamma, tissue necrotic factor alpha, and tissue necrotic factor beta, also called as lymphotoxin, and other cytokines, and of mast cell leukotrienes, which form the basis for its ability to produce toxic shock syndrome. Other T cell superantigens not belonging to the pyogenic toxin superantigen family include staphylococcal exfoliative toxins, Mycoplasma arthritis mitogen, and Yersinia pseudotuberculosis mitogen. Polyclonal activation of T cells can also occur in response to viral superantigens, such as rabies virus nucleocapsid protein. There are a number of superantigens that are capable of stimulating a substantial proportion of B lymphocytes. For example, staphylococcal protein A reacts not only with the Fc gamma region of immunoglobulin G, but also with the 15 to 50% of antibodies that utilize the variable region of heavy chain. The human immunodeficiency virus, or HIV, glycoprotein GP120, also reacts with immunoglobulins that utilize VH3 family members. This was all about superantigen stimulation. In our next session, we will discuss why do alpha-beta T cells need to recognize antigen in such a complex way.